Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Victoria Orphan. She's a James Irvine Professor of Environmental Science and Geobiology at Caltech. We're going to talk about the ecology of microbes and the different minerals and elements and things that they cycle: carbon, sulfur, et cetera, et cetera, et cetera. So, Victoria, thanks for coming. How are you doing?
I'm doing great. Thanks for having me.
Yeah. So, what are you studying, and what's the context of it?
Yeah. So I'll start with a 20,000 foot view of sort of what I view as our mission in the lab, and that is really to understand how microorganisms function in natural environments and the types of interactions that are going on between the undomesticated organisms that exist in the environment, and really how these organisms influence the cycling of globally important geochemical systems like carbon, nitrogen, sulfur.
Okay. So where do you, there's a big world out there. Where do you study them? In the oceans or, in what environments?
Yeah, a lot of our work is focused on deep ocean ecosystems. We do a lot of work in marine sediments and environments that we would consider to be extreme. So these are places where we have methane that is venting to the seafloor, or places where you have hydrothermal activities, and also even deep within the earth, the sort of subsea floor biosphere as it's called. And so trying to understand how microorganisms make a living deep in the earth.
Well, I just visited Yellowstone. They have these geysers with all these different colored rocks, and they talk about the microbial mats that are there, and that was more accessible, it sounds like, than the places that you work. But it was really cool, to see all the different colors, and you can't. I didn't have a microscope with me, but it was pretty cool to see the microbes at work. Somewhat. So
I think if you asked any microbial ecologist, they would name Yellowstone as sort of one of the playgrounds for understanding the incredible microbial life that exists on the planet, and it's pretty exciting that you can actually visually see all of the diversity through like these colors and the mineral transformations that are going on at crazy temperatures and pHs. So it's a cool place. Yeah.
So have you been able to visit some of the sites you're talking about? Have you been able to go to a black smoker or any of these places in the deep ocean?
Yeah, that's actually one of the things that I love the most about my job is, I get to play explorer as much as research scientist. And so we do these extensive oceanographic expeditions out to visit the sites, and we use human operated vehicles or remotely operated vehicles to be able to see the ecosystems that we're studying many miles below the ocean surface and collect samples. And so it's pretty exciting to be able to go down in submersibles like the Irvine with my students and sort of experience this amazing habitat that very few people have had the privilege to see.
Oh, wow. So how far down have you been? Have you gone to Challenger Deep, the Marianas Trench, or where have you been?
No, I think a very few lucky people have visited the deepest parts of the ocean, but I've been pretty deep. So I think my record is 3,200 meters depth, and that was a hydrothermal vent system off of the Pacific Antarctic ridge, so heading towards Antarctica. And was an unexplored hydrothermal vent system that we had a chance to visit and learn about sort of the life, animal life and microbial life at that site.
Oh, nice. So when you visited some of these features in the ocean, what did you see? What did looking at them for, maybe a half hour or however long. Like, what did you see that you didn't know before, just visually? Anything?
Yeah, we've been doing some work off of Costa Rica recently, which has been pretty exciting. There's a whole series of seamount systems that are venting methane. And in these systems, one of the most remarkable animal discoveries were these organisms called Yeti crabs. And these are basically they're crabs, but they have a symbiosis with sulfur oxidizing bacteria. So they have this furry white coat on them. And in order to feed their symbionts, they wave their arms over this reduced sulfide-rich fluids that are coming out of the seabed. So they're basically all dancing on the seafloor in these little congregations. And so to come upon this for the first time and watch this amazing activity going on that in darkness normally was pretty entertaining for us to observe. And just by seeing their behavior, we learn a lot about how that association occurs, and that's really an important piece of being able to visit the seafloor and see things in its natural habitat rather than just bringing samples up onto the ship.
So in this instance, the crabs are waving their arms and what, so that the stuff that's being emitted from the vents coats their arms and then other bacteria feed on it, or how does it work?
Yeah, so they are like farmers, so they actually eat the bacteria that they're cultivating on these CT that they coat their arms, and so they grow their fur coat that's made up of these sulfide oxidizing filamentous bacteria, and those bacteria need sulfide from these reducing fluids and also oxygen that's in the overlying seawater. And so the crabs basically are allowing the microorganisms to come into contact with both the oxygen and the sulfide, and they can grow these dense fur coats that then they feed on later. So they basically are growing their own food on their bodies.
Oh, wow. Oh. And eating it later. Really weird. Yeah.
It's a pretty neat trick. But I mean, the deep ocean is full of these kind of surprising associations between animals and microbes, and microbes that are also doing unique chemical transformations and physical transformations that we've only begun to scratch the surface of right now.
So what are you trying to figure out? Are you looking more at how the macroorganism, like the crab, interacts with its microbiome, or are you focusing more on the microbiome and looking at how that interacts and what chemicals it produces and metabolizes? Like, where's your focus?
Yeah, a lot of our focus are much more on the micro scale. We have the benefit of seeing all of these amazing macroscopic manifestations of microbes interacting with chemical fluids in the seafloor, but really what we're interested in are the microorganisms themselves that live in the sediments that specifically are involved in the transformation of methane through a process called anaerobic oxidation of methane. And so just like the sulfur oxidizing microbe can form a symbiosis with the Yeti crab, we're looking at microbial symbiosis that is occurring between two different types of microorganisms. So an archaea and a bacteria that uses sulfate from seawater. And so they form these nice structured communities and then collectively basically remove methane that's coming out of the seafloor using sulfate in the overlying seawater. And in doing that, they convert that sulfate into hydrogen sulfide that then becomes fuel for things like the sulfur oxidizing bacteria that are growing on the Yeti crab. And so everything is sort of intimately connected all through this chemical trophic structure in the seabed. And we're interested in the process, not just because it provides food for the Yeti crab, but also because methane is a potent greenhouse gas, and we need to understand where the sources and sinks are coming from the ocean in order to better understand what ends up ultimately getting into the atmosphere where it can cause warming. And so a lot of the implications for our research is related to how microorganisms sequester that methane before it becomes a problem through greenhouse gas effects and warming effects in the atmosphere.
At what scale? I mean, can you calculate the scale at which they're sequestering methane?
Well, so it's poorly constrained because we don't have even a good handle on all of the places where methane is venting from the seabed. It seems like the more we explore, the more we find, but it's thought that it's pretty pervasive all along the continental margins. So, and sort of rough estimate calculations suggest that this single microbial process of anaerobic oxidation of methane can remove upwards of 80% of the methane that's coming out of the seabed. So it's a pretty globally important process, and it's being catalyzed by microorganisms that we don't have a lot of understanding about in terms of being able to grow them like many of the common domesticated microbes like E. coli in the lab. So they're coming from...
Have you even tried to culture some of these, or the conditions are so different that you just know it wouldn't work?
Yeah, so this has been like a labor of love of a number of labs now, is to try to get these organisms into culture. And actually there've been some recent successes in getting them into enrichments, but nobody's been able to grow them in pure culture. So this symbiosis with the bacteria is still required, and it's hard to tease that apart. And so you get this enrichments of these organisms, but not at a level where you can get a single pure colony of these organisms right now. So that adds to the challenges in understanding their physiology and trying to understand the symbiosis, and has led to a whole assortment of sort of creative methods that people have applied to try to tease apart the secrets of how these guys are sharing energy and making a living on methane.
Is this, is there a fixed playbook of metabolism? Once you figure out how anyone can use methane, you can get an idea of how these guys do it. Maybe there's an accessible spot where creatures are, oxidizing methane, and then there'll be similar methods to the way these guys do it, the Yeti crabs, people in other inaccessible places. Maybe that's a way in.
Yeah, there is definitely something to be said for trying to find organisms that are maybe faster growing or come from a slightly less extreme environment. And if there's enough similarities in terms of their metabolic pathways, you can use that as a model and a guide. And certainly some of our most successful model organisms in the lab sort of represent those kinds of those systems. And so there are some methane oxidizers that grow in more shallow ecosystems, or they grow in places where the temperature is warmer. So there's some organisms around high thermal vents, and when you increase the temperature, you also tend to increase the growth rate of the organisms, which helps with getting them into the lab setting. And so some of the best successes we've seen now are some of these thermally adapted methane oxidizers. So you're right in, you have to selectively pick where you're going to go after trying to culture these guys.
So have you, sequenced Yeti crabs? Have you looked at the shotgun genomics of the bacteria? Have you, how much has been done to understand their environment, at least if you can't recreate it?
Yeah, we do a lot of applications of what we call sort of omics techniques for looking at microbial communities in the environments. And certainly with sequencing technology improving and becoming more cost effective, this is becoming sort of bread and butter for a lot of microbial ecologists that work with environmental systems. So we can learn a lot about the potential metabolisms of microorganisms by reconstructing their genomes directly out of environmental samples. So it used to be that we had a very narrow view of diversity of microbes in the environment through what we could culture. And that turns out to be on the order of maybe like 1% of the true diversity that exists in the environment. And we were missing entire phyla of microbial life. You can think of it like not knowing that there were mollusks out there, so no clams, no snails, no octopus, until you were able to see them through the lens of looking at their DNA in the environment. So it's a pretty exciting time just in terms of discovery of microbial life. The more we're able to apply these sequence-based methods, the more we're learning just in terms of the diversity that exists on our planet. But the true challenge then is, how do you take this blueprint that's given to us through looking at the genomes and actually then trying to understand the true function of these organisms in the environment and the kinds of interactions that are going on in between different types of organisms in the absence of being able to grow them in the laboratory. And so that's been really the big focus of my group is how we can apply some of these culture independent techniques that come from the geosciences, using things like stable isotopes to try to trace what these organisms are metabolizing in the environment and how much of that is dependent upon different organisms working together to be able to break down substrates, as we see with the anaerobic oxidation of methane.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Could you recreate any of this in the lab? What if you got like you made a special tank. It was like a 6 foot tall cylinder, clear, and you bubbled methane in at the bottom, and you try to encourage the growth of methane loving bacteria at least.
Yeah.
If you could recreate, and put a heating element, and maybe you could recreate something like that.
Yeah, you're not too far off. Actually, we are able to keep these organisms in their environmental samples going in the lab for many months at a time. And one of the things that works in our favor for studying anaerobic oxidation of methane is that the organisms grow very slowly. So their doubling times are sort of on the order of every 3 months, which means that, it's challenging to run the experiments, but they also can be maintained in the sediments as long as you give them, the basics that they need. So if you provide them with enough methane and enough sulfate, they'll continue to go. So we've been maintaining them in sort of static incubations, and we also now have just recently started trying to recreate the high pressure environments of the deep sea. So we have these large pressure vessels to see if we can sort of goose their metabolism to grow a little bit more as they might see in situ, where the methane concentrations are much higher than what we're able to do in the lab under just atmospheric pressure.
So what does the chemistry look like? Do they start with methane, and what's the end products?
Through this anaerobic oxidation of methane coupled to sulfate, the end products are CO2 and hydrogen sulfide. We now know, though, that these methane oxidizing organisms can also couple methane oxidation to other electron acceptors. So there's a group of these organisms in freshwater environments that can couple to nitrate. And there's a set of organisms that are less well studied that can couple their metabolism to metal oxides. So things like iron and manganese. And so it's our understanding of all of the different types of ways that organisms can eke out a living coupling methane to different energy sources is still expanding right now. But it seems like if you can make a thermodynamic prediction of a potentially favorable coupling, chances are we're going to find it in the environment eventually.
Okay. Actually, with that thought, are there any other candidate, chemicals that could support life that we don't know about yet or we haven't observed?
I can only tell you through the lens of methane utilization, and I can, in terms of other energy sources or redox couplings, I think arsenic is one that is predicted to occur that hasn't been shown yet. Selenate might be another one. And so there's more sort of exotic and maybe less sort of less well distributed on the globe potential electron acceptors that might also support methane-based life. And so those kinds of exotic interactions, I think, are definitely of interest in thinking about the full menu of ways that microorganisms can tap into energy, and that might have been more important through Earth's history, and also starting to think about the range of metabolisms that could support life potentially off of Earth. If we think about some of the NASA missions that are coming up, certainly it's part of the great thought experiment for researchers who study organisms in extreme environments to start thinking about what might constrain where life could exist off of Earth.
Yeah, I was going to ask you if you're contacted ever by planetary scientists, if they want to know if life is possible, in the underground oceans on various planets, et cetera, or under different conditions.
Yeah, it's one of the advantages of being at Caltech is this close connection to the Jet Propulsion Laboratory, JPL, and there's certainly a lot of cross talk between the scientists up at JPL and folks in the geological and planetary science division at Caltech. And certainly as these missions are taking more of a life-centric focus or thinking about habitable places, both on Mars and also ocean world environments, sort of Europa and Enceladus, there's been a lot of conversations and white papers being written about what to look for. So what sort of biosignatures might exist? What are the possible metabolisms? So it's becoming much more of a common topic of conversation than say 20 years ago, where it was kind of an interesting thought topic, but not necessarily with the possibility of an actual mission to look for this. So it's an exciting time. Yeah.
What do you think you're going to be able to shed light on, in the next 6 months or a year? Anything that you're on the edge of figuring out?
Well, we've been, from looking at anaerobic oxidation of methane, we have recently been very interested in how the two organisms are engaged in this symbiosis. And for many decades, the assumption was this was through a diffusible chemical intermediate that was passed from the methane oxidizing archaea to the sulfate reducing bacteria. A few years ago, we started to develop new evidence that maybe that there was a much more intimate physical connection where electrons were passed extracellularly from the methane oxidizing archaea to the sulfate reducing bacteria. So not a diffusible chemical, but rather direct sort of electrical connections between these organisms. So that was pretty exciting. And we've been continuing to explore that as a viable mechanism that allows these organisms to work together in combining methane with sulfate.
Quick question here. Do you think that, right now when we burn natural gas, which I guess is methane, we get all these combustion byproducts, it's aerobic. Do you think there may be a way figured out by studying the bacteria how to do it anaerobically and make it favorable and, make it an industrial process that uses bacteria?
Yeah, it's a great question. And there are definitely a number of labs that are interested in trying to learn from what we've discovered in looking at these natural methane oxidizing communities, about their specific genes that are involved in the methane oxidation process, with the potential maybe of genetically engineering maybe faster growing organisms to do these processes in a more industrial setting. So yes, I think there are important industrial applications that might take advantage of the sort of the genetic chassis that are in these slower growing organisms. But I think it would be hard maybe to use the organisms themselves and be able to scale it up to an industrial process, just given what we've learned about their physiology so far.
Well, yeah, so that was back to an earlier question. Are there other bacteria that are similar, that are methane loving, methane oxidizing, that are in accessible environments or might be candidates for industrial use in like a digester?
Yeah, there are other organisms certainly that can make a living on methane. Many of those use oxygen, and instead of doing this through an anaerobic metabolism. So they call them methanotrophic, mostly bacteria. And those organisms are being used in a variety of different contexts right now. And they tend to grow a little bit faster. And there are cultured representatives of them. So it's a totally different set of metabolic machinery that they use to oxidize methane coupled to oxygen. So it's much more energetically favorable than these archaea that are doing it through an anaerobic pathway, where they're getting just a fraction of the energy that you would be able to recover with oxygen.
Have you looked for phages that interact with the archaea?
Yeah, that's a great question. We have a new program in the lab that's focused on looking at viruses and bacteriophages in these methane seep ecosystems. And we have some sort of early indications that there are viruses that are connected to these methane oxidizing archaea, but it's still early on. And, as challenging as it is to study uncultured bacteria and archaea in the environment, you can add another layer of challenge to try to understand the viruses that are infecting them, because it's when you use metagenomic types of techniques to look at the diversity of viruses in these environments, most of that data comes back as completely unknown. So we've barely begun to scratch the surface in terms of the diversity of phages out there, and it's going to take a while to start linking that sort of unknown gene space to actual viruses that are infecting the organisms themselves.
Okay, well, very good. Well, Victoria, what's the best way for people to find out more about your research?
Yeah, best place is to look at our website. So we have Orphan Lab, www.orphanlab.edu, or you can find me on the Caltech Geobiology option webpage at Caltech.
Okay, well, very good. Victoria, thanks for coming on. I appreciate it.
Yeah, it's wonderful talking to you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.